Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligands. Hyperlab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligands. Hyperlab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. Hyperlab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. Hyperlab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. Hyperlab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. Hyperlab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. Hyperlab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. Hyperlab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. Hyperlab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics.
Hello everyone, it's time to start. I'm Jetang Lim, CTO of HIS. Thank you so much for joining us. The title of today's webinar is How Deep Learning is Transforming Drug Discovery. Let's take a quick look at the agenda first. In the first part, I will talk about what it means to learn. Then in the second part, we will look at how learning connects to deep learning, focusing on the key methods in AI. After that, I will discuss why deep learning, which is our main theme, is so well suited for biostrategy development. Next, I will go over our upcoming webinar schedule and take some time to answer questions we've already received. Finally, we will have a live Q&A session where I will answer any questions from you. So as you listen, if any questions come to mind, please make a note of them and share them at the end. I hope this will be a helpful and engaging session for everyone. So now let's move on to our first topic. The question, what does it mean to learn, is quite philosophical. There are many ways to answer this, but today I will focus on answering from a neuroscience perspective, looking at what learning means from the standpoint of our brain. Before that, let's consider one situation. Imagine, as shown in the picture, that we are drinking hot coffee. And this process of drinking hot coffee is actually very complex. If instead of an adult, we imagine a 3 or 4-year-old child trying to drink hot coffee or tea, then many concerns may come to mind. We might worry about them dropping it or burning their mouths. However, others perform this action of drinking hot coffee quite naturally, even several times a day. And not only we can drink coffee, but we can do so without full concentration. We can listen to music playing in a cafe, engage in deep discussions with colleagues, and still drink the hot coffee almost subconsciously. We can do this while simultaneously smelling the aroma and savoring the taste. If we asked a robot to perform this complex task, it would be extremely difficult. The principle lies in repetitive learning. The repetitive learning enable us can perform various actions that require high concentration unconsciously, as shown in the image below. For instance, Hyunjin Ryu, his impressive cutter strike in Major League Baseball comes from the countless repetitions of training. Similarly, when Son Heung-min accurately scores a goal from the Son Heung-min zone in a dynamic situation, it's also the result of countless practices in that zone, and it leads to an almost unconscious action. And Tiger Woods' iron shot would be a similar result. But when these professional athletes perform these actions, they do not calculate the exact angle, speed, and spin needed to hit the target accurately. They just think, I should throw like this at this moment, and the rest of the action happens automatically. So this shows that through repetition, these highly concentrated actions become ingrained in the brain. So repeated training develops intuition, and through expert intuition, one can make excellent judgments in various situations immediately. And this is not limited to sports, but has also been proven in various fields, such as medical knowledge or a firefighter's ability to make instant decisions on site. So the repeated training builds excellent intuition in these areas as well. Dr. Anders Ericsson expresses this as the 10-year rule, stating that if you invest 3 hours a day for 10 years in a specific field, you can become an expert in that field. In terms of time, this is about 10,000 hours. Another person, Malcolm Gladwell, calls this the 10,000 hour rule. To give a more interesting example, developing new drugs or researching biotech in a specific academic field is also an expert domain. If we assume it takes about 5 years to get a PhD, working 40 hours a week, it amounts to about 12 hours. And this is quite similar to the 10,000 hour rule. However, it may take even longer for like in drug development because it requires much deeper expertise than other fields. Thus, numerous repetitions give our brain the power to develop intuition. Let's see what exactly happens in our brain. The most basic component of our brain is the neuron. This image shows the structure of neuron, which I'm sure everyone here is familiar with. The neurons have dendrites that receive signals. When they are activated by chemical signals in the axon, they transmit electrical signals. And this process is repeated as the signal is passed to neighboring neurons through synapses. And neurons receive input and send output with the signal converted to electrical signal by the movement of ions. And this signal is analog, and chemical neurotransmitters are transmitted at synapses. And this can be represented as an activation function. Then as time passes, when specific neuron receives a signal, If the signal's magnitude exceeds a certain threshold, the neuron is activated. Otherwise, it remains inactive. Through this process, analog signals are converted to digital on-off signals. Then when drawn as a function, this can be called a sigmoid function or logistic function. If a specific input value is greater than 0, it produces a value close to 1. Otherwise, it produces a value close to 0. Thus, converting continuous input values into digital signals close to 0 and 1. Such complex processes of intuitive judgment is not possible with just a single neuron. In reality, our brain has countless neurons. For example, an adult brain is said to have about 1 trillion neurons. Each neuron is connected by an average of about 7,000 synapses, and some can have up to 10,000 connections. And through this complex synaptic network or neural network, we can perform the process of learning. Importantly, once a neural network is formed, it doesn't remain static, but it changes in response to external stimuli and making it an adaptive system. Every time we see, hear, or experience something, our brain receives stimuli, and accordingly, the strength of synaptic connections can change, and as a result, new synapses can form or existing ones can be disconnected. And through this process, the magnitude of signals changes, and therefore in this adaptive system, it is important that it changes in response to external stimuli, and this is what creates our ability to learn. And based on learned information, we can make accurate inferences when throwing a ball like Yuhyunjin, as I showed in a previous slide. And this is how new abilities emerge in an integrated system that cannot be explained by individual elements alone. We call this an emergent phenomenon, and the brain is the most representative example of such emergent intelligence. The brain continues to change even after reaching adulthood, which we call neuroplasticity. Brain plasticity refers to the ability to change through learning and experience, which is essential for learning new skills or forming memories. For example, when you see a cat on the street or in a photo, light reflected from the cat forms an image on your retina. The signal from the retina initiates neural activity through the optic nerve, and as that signal is transmitted to other neurons and reaches specific areas of the brain, the recognition of seeing a cat occurs. While this process happens very naturally and quickly for humans, traditionally replicating this through programming has been really difficult. The process known as perceptron was particularly challenging to program. However, today such pattern recognition tasks are being performed much more easily through artificial intelligence, specifically deep learning, which I will explain. As I mentioned earlier, the brain forms the complex neural networks with countless neurons, providing various leading abilities. And this works when we learn to drive, cook, study various academic fields, or develop athletic abilities. The brain has a very versatile system, and in this sense, our brain can be called a universal computing framework. In some situations, physical laws can be very useful, while we can develop intuition through repetitive training when learning. We can also predict things through physical laws like F equal ma or mathematical reasoning. For example, when predicting the trajectory of a rocket being launched to Mars or its return to orbit, training is not possible. It's too costly and time-consuming. In such cases, using physical equations to calculate precisely with computers allows for more accurate predictions. So in next part, we will see how learning connects to deep learning. So let's explore that now. As explained earlier, we have neurons, and if we create a mathematical model of neuron to be processed by computers, we can make virtual neurons and even a virtual brain. This idea started in the United States in 1957, leading to the proposal of the perceptron, an artificial neuron. Like the brain's dendrites, the perceptron has input channels that receive signals, and it has weights representing the strength of synaptic connections, as shown in the figure. And by multiplying this, we can calculate the sum of signals received by a specific neuron. Then by adding a unique bias value to each neuron to determine the total signal strength and putting this value into an activating function like the segmented function we discussed earlier, then the final output is produced. Through this, we can create a virtual mathematical model which we call an artificial neuron or perceptron. The next step is to complexly connect these artificial neurons, just like our brain. Each of these dots you can see represents an artificial neuron, and the lines are synapses connecting neuron to neuron. And we can mimic the brain's function by multiplying each line by a weight value to represent the strength of the synapses. We call this connected network a deep neural network, and this deep neural network is the core architecture of deep learning. And through deep learning, we've been able to solve a variety of problems. I will show you one typical example. There was a competition called ImageNet Challenge. The challenge is to distinguish images of dogs and cats. And this competition had been around for a while, but until 2011, classic machine learning method, which is not deep learning, performed better than deep learning. The results of this competition were measured by the error rate. The error rate is how many mistakes were made when 100 photos on an average is shown. With the best performance at that time was about 25%. However, from 2012, as deep learning began to be applied, the number of hidden layers gradually increased, the performance dramatically improved. By 2015, the error rate had fallen to below 5%, and the competition became meaningless and was terminated. And it showed that the more complexly we design deep neural networks like the human brain, and the more we stack them, the more rapidly performance improves. And this is the improvement of learning ability. And there is a reason why deep learning works so well. It's called the universal approximation theorem. It shows that we can approximate any nonlinear function by stacking enough neural networks. This theorem mathematically supports the idea that deep learning is a powerful tool capable of solving very complex problems, very complex nonlinear problems. For example, when we want to predict the properties of a drug, we might want to input the drug structure and get output related to properties such as toxicity, solubility, and the drug effectiveness. In these cases, the relationship between input and output is mostly complex nonlinear, so deep learning can approximate this complex relationship very precisely based on the data. Also, the deep learning can be interpreted from the perspective of probability learning distributions from the data. For example, let's say that we have various image data of cats, and the deep neural network learns the probability distribution of these data. Therefore, if a particular cat photo is likely to appear frequently, then then the likelihood of generating that image increases, and conversely, a rare cat would be generated with low probability. And by learning such probability distributions from the data, the deep learning network can even generate new cat photos. And it works similarly with language problems. It operates by probabilistically predicting the next word or alphabet in a given sentence. For example, when given a sentence like "I like blank," the deep learning model predicts the next word according to the probability distribution. If the probability of "I like science" is 5%, the model will recommend it with a 5% probability. And if "I like cats" also has a 5% probability, it will be predicted equally. If the expression "I like hyperlapse" appeared with a 90% probability, the deep learning model would choose hyperlapse with a higher probability than previous 2 hours. And this is how deep learning operates based on probability distributions. So based on these principles, deep learning works in various fields that we can actually experience. A representative area we are already familiar with is language. Deep learning solves language problems very well. For example, when we see a simple sentence like "Hello, nice to meet you," our brain immediately understands what it means. Similarly, deep learning model learns such sentences to translate or comprehend their meaning. However, if we don't know Arabic, we wouldn't be able to understand the meaning of a sentence. Likewise, if deep learning hasn't learned a new language like Arabic, it would struggle to comprehend it. So deep learning makes tasks like translation possible by learning the probability distributions of various languages. So the ability of deep learning to learn these probability distributions from numerous data is utilized not only in language, but also in image, video, games, autonomous driving, robot control, weather prediction, and more. So, for example, by learning the vast amount of video data existing on YouTube or the internet, the deep learning can build a system that can predict and recommend videos that you want. So same principle also applies to Go. Go can be seen as a problem of probabilistically predicting where to place the next move after the first move. The humans also rely on intuition when playing Go, and deep learning develops such intuition by learning from numerous data. For example, Sadoli was the best Go player at that time, and it's estimated that he played about 10,000 games of Go. However, AlphaGo played about 100,000 times of games in a year, which is equivalent to 200 years of human training. In other words, AlphaGo had more than 10 times the training of Sadoli, so it wasn't surprising at all, considering this huge gap in training. So later, DeepMind developed a new model called AlphaGo Zero, which learned Go through self-learning without looking at human game records. AlphaGo Zero beat the existing AlphaGo version in just 3 days of learning, and in 21 days it surpassed the AlphaGo Master version that had defeated Koje, the Chinese Go player. After that, AlphaGo stopped developing further, and it is considered to have reached a godlike level. Moreover, deep learning can process much more data than humans. For example, the humanoid robot developed by OpenAI listens to human language commands and recognizes situations through reason accordingly and perform appropriate actions. When a person says, "I'm hungry, give me an apple," this robot can accurately pick up an apple from the table and deliver it to human. It can also perform tasks like cleaning up the table when commanded. In the past, it was impossible to command robust to perform such complex tasks. But now through deep learning, we can perform these tasks without complex calculations. And deep learning is also very useful in weather prediction. Recently, an AI called GraphCast, developed by DeepMind, can predict 10 days of weather in less than a minute. In the past, these tasks took a very long time, even with supercomputers, but GraphCast enables faster and even more accurate predictions. In fact, this AI was evaluated to be over 90% more accurate than the European Weather Agency's supercomputer. Similarly, it has been reported that AI-based weather forecasts are now possible even with home computers, not supercomputer, and this can provide more accurate forecasts than supercomputers. And it shows how deep learning is influencing various fields in our daily lives. The fields where deep learning is used, such as language, image, video, games, autonomous driving, robot control, weather prediction, are continuously expanding, and this is similar to how our brain learns and executes various things. So far, I've explained the basic principles of deep learning and its application cases. So now, based on these principles, I will discuss why deep learning is suitable for biodrug development. So when we talk about learning, we can think of two main approaches. One is predicting based on mathematical and physics principles, and the other one is predicting through intuition. For example, when predicting the trajectory of a rocket, physical laws are very accurate. We have very strong physical method. In such situations, calculating through mathematics is more appropriate. However, for more complex problems like weather prediction, driving, speaking, or playing Go, intuition often works better. The more complex the problem, the harder it is to solve with physical approximations. So an intuitive approach often fits better than physical approaches. In these situations, deep learning can work very effectively. So biosystems, as you know, are very complex, and in the biosystem, various entities like DNA, RNA, and proteins work on multi-scales, and each component interacts with others to create the functions of the human body. The human body has about 60 trillion cells, and within them there are numerous proteins, and these interact with each other to form complex systems. And even small changes like point mutations can have a big impact on the overall function of cells. So we can say that human system is non-linear complex system, and these complex systems present problems that are really difficult to be solved with physical approximations. So the more nonlinear and complex the system, the better deep learning works. Within complex biological systems, there are numerous cell-to-cell variations, which also leads to large variations between species. And biosystems are fundamentally composed of multimodality, and it results in emergent phenomena where each entity interacts in complex ways. Because each cell and person is different, a statistical analysis is necessary, and because reactions are nonlinear, they are difficult to predict with physical approximations. For these reasons, deep learning is very suitable for these complex systems. In fact, there have been successful applications of deep learning in the biofield. The most representative example at the molecular scale is the protein folding problem. So deep learning solved this problem in just 5 years, which couldn't be solved by the physical method for the past 50 years. Deep learning solved this problem by learning data distributions, and this is considered one of the most innovative success in bioresearches. So Clarivate, a paper study site, predicts that Nobel Prize candidates every year, and this year, Demis Hassabis, who developed AlphaFold and AlphaGo, emerged as a strong candidate, and indeed, Hassabis received the Nobel Prize in chemistry. AlphaFold was recognized for its contribution to computational biology, that it helps to understand the protein world and publicize the remarkable research results of experimental biologists. In other words, it means that AI is key evidence that will make science faster and ultimately help understand disease and develop treatments. So we've already experienced success at the molecular study, and we can further expand to more complex gene level or cell level. I'd like to emphasize that it's very early stage of the research. One example, a paper introduced in Cancer Cell in 2020 used gene ontology and drug response data to apply AI to predict drug responses and the synergies in the human cancer cells. And this study used two important inputs. The first one is gene type of cancer cells, and the second one is the drug structure. And by combining these two, it predicts the drug response in cells through deep learning. So the researchers received evolution type data and included point mutation information to increase the interpretability. And based on this information, they created a hierarchical tree structure and embedded signal transduction pathways, cell-level processes, and molecular structures into deep neural networks to ultimately predict drug responses. The researchers didn't just create the model, but they proved it through experiments. For example, when a specific drug was treated to cancer cells, AI predicted the target pathway and discovered new pathways and predicted synergistic effects. When this synergistic effect was verified through CRISPR technique, it was confirmed that the predicted target pathway indeed had a greater impact on cancer cell death. And furthermore, the researchers created a xenograft model from samples of about 400 clinical cancer patients and experimentally verified the synergistic effect when 6 drugs were treated in combination. And this model showed that synergistic effect actually existed as predicted by AI and could extend the patient's survival time. And through this, we can confirm that deep learning can work successfully even at the gene scale or cellular level. Of course, we should note that this research used well-refined data like ontology. According to a recently published paper, a bold goal of creating a virtual cell has been proposed, and this paper deals with how to create a virtual cell in a data-driven way with a physical approximation. To achieve this goal, multi-omics data is needed, along with AI and computational power. AI and computation power of processing it. If these elements are in place, we can implement from molecular level to cellular level and further to tissue dynamics. Through them, we can develop personalized cells or personalized medicine. And this paper explains the process of various entities at the molecular scale gathering to interact and create functions at the cellular scale to simulate actual cells. And it suggests that how cells interacting at the cellular scale change and react at the tissue level can be simulated through data-driven AI. Currently, many architectures have been developed that can well represent DNA, RNA, and proteins through deep neural networks. For example, transformers are very useful for processing such data and can analyze perturbation data or gene expression profiles using omics data. Orthographic neural networks can be used to express cell-to-cell interactions, and diffusion models can be used to simulate timescales. Various tools like these are available, and through them we can solve complex problems. But it is still quite challenging to apply deep learning as successfully in biodrug development as in natural language processing. One main reason is data. For example, the short-read archive, a sequence database, holds 14 petabytes of data, and this is more than 1,000 times larger than the data used to train ChatGPT. And nevertheless, the biofield is not showing as much progress as demonstrated in natural language processing. And this is because cell fusions are much more complex than natural language. Cell-to-cell interactions are very diverse and they differ for each person or species. As biosystems deal with such diverse and complex data, there are limitations in considering all of this. And to solve this problem, we are currently taking an approach called reductionism. That is, rather than simulating the entire cell, we distinguish each element and try to predict by understanding each element. And this approach has been partially successful, like AlphaFold, and AI models predicting cell responses continue to develop. So, however, a reductionist approach cannot fully simulate the new emergent phenomena created by aggregate. To solve this, we need to well integrate the models completed for each element to create an integrated universal model. We haven't reached this goal yet, but the vision of virtual cell presents the possibility of achieving this goal. Currently, we can say that we are at the very first stage of drug development AI. For example, in the drug development process, AI is being used in various experimental processes such as cell-free, cell-based, ADMT, PK, and etc. In HyperLab, we are also developing AI models suitable for each task and performing prediction tasks for each task. However, there are still some limitations in fully simulating emergent phenomena. One fortunate fact is that the data is increasing rapidly. Currently, there are more than 100 million single-cell data, and this data is doubling every 6 months. As so much data accumulates, the data problem when access of deep learning can be somewhat resolved. Of course, learning such a large amount of data is another challenge in itself. So this is the end of my talk. And now in our fourth quarter webinar, we plan to discuss how we can advance AI algorithms in situations with insufficient data. In particular, we will take a closer look at foundation models and discuss how this foundation model can contribute to solving complex problems. And next, there were a few questions that came in advance, and I'd like to answer 2 of them. The first question was about how computational techniques are applied in lead optimization and whether the lead optimization stage is generally conducted only by pharmaceutical companies. To answer briefly, as I showed in the diagram earlier, in the drug development process, moving from the early stages to later stages includes the lead optimization phase. In reality, is a bit more complex and computers are actually used. And as I mentioned earlier, for individual tasks, we predict things like plasma stability or half-life. We just can't integrate all of this together. So in reality, these predictions need to be verified through experiments. And predictions and experiments are learning concurrently, like Penrose's staircase or in a human-in-the-loop format. This is how AI's shortcomings are complemented by experiments. And conversely, AI complements the trial and error that could occur if we rely solely on experiments. And the second question was about accuracy of properties AI, properties predicted by AI, and the success rate of discovering effective substances, as well as how similar these are to actual experimental data. So this actually varies by project, but according to our test in a paper we published in 2024, this year, we plotted the correlation coefficient between predicted and experimental values for various target proteins and their known risk substances. And for each target, as I mentioned, the correlation coefficient varies from over 0.8 for the highest to the under 0.4 for the lowest. And there's difference in performance. Then we show this result for 8 targets, and the average value is about 0.64. So the model we developed performs quite well compared to other models, and it is more than twice as good as commercial software like G software and non-commercial docking tools like V software. And it's similar in performance to F software, which is MD based, but about 10,000 times faster in computational time, since molecular dynamics is really slow. So we can do these things quickly and accurate through AI. And post-prediction is also one of the most important problems. And there are various deep learning models and docking models. And this is the method we created. And this is AlphaFold. This is our method, and this is AlphaFold. And as you can see, AlphaFold is the most accurate. And although we are not quite that accurate, but we are much better at predicting poses than other models. Then what I showed earlier was for non-covalent bonds, the result for non-covalent bonds, and we can also apply similar models to covalent inhibitors. And there aren't many other models for this. We started this project this year, and when we started in March, we started with only about 3,000 experimental data points, and the correlation coefficient was only 0.2. But as of July, it has improved to 0.77. It's quite a significant increase, and we continue to train to show higher performance. And let's see in actual experiments. I'm not sure about the performance of other tools, but here's an example of an internal experiment that we conducted through a CRO using HyperLab. That we screened 1 million compounds for a specific kinase target using HyperLab and selected the top 100 and filtered them. Then when we experimented with this, 9 came out as hits. 9% came, the hit ratio is 9% based on IC50 criteria. Then somewhat below 10 nanomole, and some show cellular activity below 10 micromole. While the hit criteria may vary, I think this shows quite good performance. Of course, it should be noted that it varies depending on the method and target. So with this, I will conclude the pre-Q&A session, and I will move on to a live Q&A session. So if you have any questions you want to ask, please leave a comment, and we will have a short 5-minute short break, and I will come back to answer the live Q&A sessions. So see you at 5 minutes later.
I think all you needed to do was type in the PDB code. It would get it for you, and then you can draw in your ligand. HyperLab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. Hyperlab is a lot, more streamlined in comparison to other docking methods. And then you can automatically go through and do the docking, do prediction of the different physical characteristics. Yeah, so Hyperlab definitely made, everything a lot more streamlined, a lot simpler. I think all you needed to do was type in the PDB code, it would get it for you, and then you can draw in your ligand. Hyperlab is a lot, more streamlined in comparison to other docking methods.
So if there are no questions, I'd like to wrap up the webinar. And if you have any questions that pop up to your mind even after the webinar, we offer a coffee chat session with AI experts for free. So don't hesitate to contact us. So thank you for your attendance and active participation in this webinar, and look forward to seeing you again in fourth quarter seminar. Thank you so much.